By employing inventive methods, researchers have successfully fabricated synthetic cells that exhibit the same behaviors as their living counterparts. These groundbreaking approaches have effectively merged the realms of artificial and organic materials, resulting in the creation of fully functional cells. Greetings, everyone. Today, we will be delving into the subject matter of a recent study featured in Nature Chemistry. The study, conducted by Ronit Freeman and her team at UNC Chapel Hill, explores the groundbreaking techniques they employed to manipulate DNA and proteins, the fundamental components of life. Their remarkable achievement involved the creation of cells that closely resemble and function like cells found in the human body. This pioneering breakthrough holds significant promise for advancements in regenerative medicine, drug delivery systems, and diagnostic tools. Without any more delay, let us begin our insightful conversation. Freeman, whose lab is located in the Applied Physical Sciences Department of the UNC College of Arts and Sciences, envisions a future where engineering fabrics or tissues can adapt and respond to environmental changes thanks to this groundbreaking discovery. The building blocks of cells and tissues are proteins, which collaborate to carry out functions and construct structures. Crucially, proteins play a vital role in the creation of the cell's framework, known as the cytoskeleton. Without this framework, cells would be rendered incapable of functioning properly. The cytoskeleton grants cells the ability to adapt and remain pliable, allowing them to adjust their shape and respond to the surrounding environment. Using a revolutionary peptide DNA technology, the Freeman lab successfully constructed cells that possess dynamic cytoskeletons capable of adapting to their environment. By harnessing the power of programmable peptides and repurposed genetic material, they were able to create a functional cytoskeleton without relying on natural proteins. According to Freeman, it is not typical for DNA to be found within a cytoskeleton. However, through the process of reprogramming DNA sequences, we were able to transform it into an architectural substance that binds peptides together. When this specially designed material was placed in a water droplet, it began to form intricate structures. Scientists have the capability to manipulate DNA, allowing them to engineer cells with designated purposes and regulate their reactions to external pressures. Although the synthetic cells developed by the Freeman lab are less intricate than their living counterparts, they are also more reliable and less vulnerable to harsh conditions such as extreme temperatures. According to Freeman, the synthetic cells demonstrated remarkable stability even in extreme temperatures of 122 degrees Fahrenheit, thereby presenting the opportunity to produce cells with exceptional capabilities in environments that are typically inhospitable to human life. Rather than producing durable materials, Freeman emphasizes that their materials are designed for specific tasks and have the ability to adapt for new functions. By incorporating various peptide or DNA designs, their applications can be tailored to program cells within fabrics or tissues. These innovative materials can seamlessly integrate with other synthetic cell technologies, offering immense potential to transform fields such as biotechnology and medicine. According to Freeman, this study provides valuable insights into the essence of life. He asserts that synthetic cell technology has the potential to not only replicate natural processes, but also produce materials that exceed the capabilities of biology. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.